If you could design the perfect child from scratch, defying the laws of natural evolution, but in turn, providing them with gifted genetics and a plethora of strong personality traits to optimize their chance at a successful life, would you willingly take the risk to solidify your genetic lineage? This profound ethical question is being tackled today through the science of gene editing, allowing humanity to flirt with the dystopian themes of 1997's blockbuster film Gattaca, which showcases a future society driven by eugenics and the power imbalances it creates. The birth of gene editing began back in the 1970s from a scientist by the name of Paul Berg. He was the first to create recombinant DNA technology, which allows genetic material from one organism to be artificially introduced, replicated, and expressed into the genome of another organism. Even though Berg won the Nobel Prize in 1980 for his seismic discovery, its time-consuming, difficult, and expensive nature made further research into the field tedious and uneventful. After an unremarkable 30-odd years, other than, of course, Dolly the sheep being cloned in 1997, a pivotal scientific breakthrough emerged in 2012. Streptococcus pyogenes, the bacteria found in strep throat, was discovered to contain CRISPR RNA and CRISPR-associated protein 9. CRISPR was the bacteria's primary defense mechanism against viral infections. Significantly, this CRISPR molecule could be manipulated by scientists to specifically target and edit any part of the human genome, which was once thought to be unchangeable and predetermined from birth. Even in the past few years, CRISPR has gone beyond altering DNA, as it has seen use in altering chromatin and targeting epigenetic regulation. Epigenetics is just a fancy way of saying how our genes strengthen and adapt based on our life circumstances and daily habits, all of which occur, of course, after birth. Applications of CRISPR include altering defective genes, such as the beta-globin gene in sickle cell anemia, altering oncogenic genes to prevent cancer from developing, and reducing telomerase activity to prevent aging. In 2018, the world was once again shocked by He Jiankui, a Chinese scientist who used CRISPR to alter the genes of an embryo that ended up resulting in the birth of newborn twins that had protection against contracting HIV. After having been sentenced to 3 years in prison, and despite the societal backlash for this mind-altering experiment, He Jiankui returned to the lab and in a recent interview mentioned that society would eventually accept human embryo gene editing in the quest to find treatments for genetic diseases. The twins will be monitored as they continue to develop, and new research shows that the same alteration introduced into the girl's DNA, the deletion of gene called CCR5, not only makes mice smarter, but also improves brain recovery after stroke, and could be linked to greater success in school. Gene editing, which was once met with cynicism and considered a taboo topic, has now had the floodgates opened thanks to He's experiment, with the consensus having shifted from an outright ban to determining what technological advancements would be deemed acceptable from the scientific committees around the world. Testing has remained halted on human embryos for the time being, but who can foresee how regulations will change in the future? Despite the unknown risks and ethical dilemma gene editing faces, the main benefits are fourfold: disease prevention, increased yield of agriculture, the treatment and cure of diseases, and the creation of model organisms to achieve scientific breakthroughs. Overcoming all diseases would be a massive win for humanity to increase our average lifespan to centurion status and beyond. And yet, we cannot underscore the importance of the genetically edited foods and animals that contribute greater food availability as our global population continues its seemingly unstable expansion. Genetically edited foods differ from genetically modified foods, or GMOs, which dominate today's headlines. GMOs, a much older technology, involved adding new genes to plants and animals, and sometimes these genes were from an entirely different species. By contrast, gene editing involves more precise changes to the plant or animal's DNA. These small sections of DNA that become edited facilitate changes that could be produced through natural means, like traditional crossbreeding, only much, much faster. Gene-edited foods are already on the shelves in Japan, for example, such as tomatoes rich in a chemical that supposedly promotes calmness, red sea bream with extra edible flesh, and pufferfish that grow more quickly. In the US too, firms are developing heat-resistant cattle, pitless cherries, and seedless blackberries. Other countries are also pressing ahead with the plans for gene-edited crops at great speed, as Thailand recently joined Canada, Australia, Japan, Brazil, Argentina, and the USA in adopting regulations around gene editing. If you think foods are the only thing being genetically edited, think again. as animals themselves are also undergoing gene editing to reduce animal suffering, use fewer antibiotics, and even change the way methane gas is produced from livestock such as cows, goats, and deer. This could have a massive rippling effect on greenhouse gas emissions and ultimately greatly improve climate change. A prime example of a reduction in unnecessary animal suffering is how gene editing has enabled the egg production industry to produce female-only chicks to avoid the need for billions of male chicks being killed each year when they are just a day old. Not only are the genetics of animals being examined, but viruses are also a big problem impacting livestock supply chains, such as pig disease, which is wiping out pig herds in Spain, African swine fever, which is impacting Europe, and the bird flu virus that's been detected in dairy cattle in their milk in the USA. To tackle this problem, Professor Sang from the Roslin Institute, the same institute where Dolly the sheep was cloned, used gene editing to develop a strain of pig that is resistant to the pig disease. Although the PRRS-resistant pigs aren't commercially available right now in the UK, they have received regulatory approval for their use in Colombia. The impact of this technology could improve the lives of the poorest farmers, as 70 to 80% of farmers are smallholding farms with 2 to 3 animals, and a devastating disease can leave a farmer and their family with nothing. It's not all sunshine and rainbows, though, as scientists would like us to believe. For example, the Compassion and Work Farming Group's biggest fear is that gene-edited animals made more resistant to diseases will mean that the industry will not be motivated to deal with the conditions that lead to the animals getting ill in the first place, such as crowded, unsanitary conditions. Already, selective breeding tactics have brought a lot of problems over the past 50 years, as chicken, for example, have been bred to grow so quickly that their legs and heart can't properly support their rapidly developing body, leading to painful leg disorders and heart disease. Okay, so up to this point, we've examined how gene editing has had numerous impacts on the foundation of human development, animals, and the foods we consume. But what about the ethical perspectives behind the actual technology of gene editing? The most controversial of all proposed gene editing applications is germline human genome editing, the exact technique that was used on the newborn twins to protect them against HIV, as this alters the genome of a human embryo at its earliest stages. This may affect every cell, which means it has impact not only on the person who may be born, but possibly on his or her descendants as well. Proponents against gene editing believe that its usage undermines our humanness, which is our evolutionary birthright, and has been earned through the toll of billions of deaths before us, each a gift of natural selection. Philosopher Michael Sandel argues that genetic engineering erodes our appreciation of the gifted character of human powers and achievement, and weakens 3 key features critical for our long-term survival: humility, responsibility, and solidarity. This opinion ties closely with the theological viewpoint that gene editing would involve playing God and disrespecting God's creation. This, of course, contrasts to the philosophical and biomedical perspective that believes it's our duty to improve the human condition. One thing to also consider is the varying cultural and public perspectives that include differences in nationality, religion, and political affiliation, leaving a very real question mark on how each nation would use this technology responsibly should it become commercially available. A second opinion states that gene editing could actually be used as a weapon of mass destruction as biological warfare becomes more present in future generations. A third daunting scenario is brought to light by Yuval Harari, who imagines a small class of superhumans dominating an underclass of useless people. Alternatively, this underclass may become a standing reserve and an orderly resource for technical application or a mere source of energy. While this viewpoint is rather extreme, it highlights the undeniable power imbalances of the socioeconomic gap widening that takes into account prejudice versus parity, justice and equality, equal distribution to members of society, and what benefits certain groups might be a detriment to others. A final ethical debate is the very act of utilizing these technologies to cure people discriminates against people with disabilities, assuming that they need or even want curing if treatment was made possible. Along with that, if certain disorders can be treated but not others, this would incite a massive lobbying on where research efforts should be focused, and of course, typically corruption follows. Proponents for gene editing argue that evolution progresses by random mutations in the genome, which dwarf what can artificially be done with CRISPR. These random mutations often cause serious problems, and people are born with serious defects. In addition, we have been manipulating our environment in so many ways and exposing ourselves to a lot of chemicals that cause unknown changes to our genome. Through all of these potential harmful effects that our ancestors never experienced in today's bustling society, why wouldn't we utilize a precise intervention to cure diseases and increase our overall quality of life? The focus should shift not to whether this treatment is beneficial, but instead how it can be ethically regulated by governments, corporations, and the scientific community for the well-being of humanity and our continued evolution as a species. Thanks for taking the time today to learn about the science of gene editing, its impact on human development, disease treatment, food production, and its ethical concerns. Let me know what you guys think in the comments below, and until next time, please like and subscribe for more great content. Thanks.